Hey guys, this is Kendall Terry, and we're going to look today at the evolution of populations. So we've been talking about evolution, and now we're going to put some math to that to try to dictate whether we can look at populations and tell whether they are in any kind of evolutionary process or not, and then try to identify what that evolutionary process may be. So to understand this, we first have to go back to a couple definitions to remind ourselves some of these key terms that we're going to be using. For instance, population, which is a localized group of individuals belonging to the same species. Very important there. Of course, obviously looking at a particular area, that's the word localized group. A species is a group of populations whose individuals have the potential to, and these are the two key parts of species, interbreed and produce fertile offspring in nature. This is not talking about what we're able to accomplish in a lab or in a zoo or anything like that. This is saying in a natural environment, in nature, they will be able to reproduce with each other, they will interbreed, and they'll produce this key thing right here too, fertile offspring, meaning offspring that can also reproduce another generation. We're looking at a population. One of the things that we need to take into account is the gene pool, and that's the total aggregate of genes in a population at any one time. So this is looking at the actual alleles that population has and the characteristics that population possesses. So when we're looking then at a gene pool, we like to look at the gene frequency in a population. Remember, diploid organisms, which is primarily what we're focusing on for this course, have two alleles for each gene. So when we're determining then the gene frequency, we must account for each of those alleles. So let's look at an example. Let's say we have a population that has 500 plants. The characteristic we're looking at is the color of the flowers. So we have a big R representing red flowers. We have a little r representing white flowers. So then if we're told that out of those 500 plants, 20 of those plants are white flowers, that means that we know that their genotype is little r, because if they have white flowers, they have to have the recessive alleles. So that would leave them 480 of those plants to be red flowers, and that's going to be a combination of both those homozygous, dominant, big R, and those heterozygous, big R, little r. So then let's take that a step further, and let's say that we know information that we may not know if we just encounter this population in nature, and say that we know that 320 of them are big R, whereas 160 are big R, little r. So then if we're trying to, which is the whole point of this, to calculate the gene frequency in a particular population, then we can say, well, if for the frequency of big R allele by itself, then we would look at 320, which is the number of these, times 2, because they each have a big R allele, plus 160 times 1, because we've got to account for these individuals that also have one big R as well. So when we do that, we end up with 800 big R alleles in our population. Well, remember, there's 500 individuals. They're all diploid, which means they have two alleles for that particular gene. So we say there's 1,000 total alleles in the population. 800 of them are big R. So the frequency then would be 0.8. Now we could turn that around and do the same thing for the little r allele, which you would have 20 times 2 for these, plus 160 times 1 for the little r here in the heterozygotes, and that would give you 200, and then 200 divided by 1,000 would give you a gene frequency of the little r allele being 0.2 in this population. So let's take that then a step further. Now we look at a theorem called the Hardy-Weinberg theorem. So we've got a little basis now of frequency and gene pool in our population, but now what can we do with that? Well, we go to the Hardy-Weinberg theorem. It's named after two people that worked on this, last names Hardy and Weinberg. The frequency, this is what they said, the frequencies of alleles and genotypes in a population's gene pool remain constant over generations unless, this is a big unless, acted upon by the agents, and agents would be a number of things. We'll look at what those are. We'll actually define those in a minute. Other than Mendelian segregation and recombination. And this is saying these are the two that just happen by chance anyways, We've talked about that before with genetics. So other than these two things, if there's anything else acting on them, that's going to evolve that population. If this is the only thing happening, then we say that population is in equilibrium or is not evolving at this current time. So let's go back to that previous example. So in the previous example, the big R allele frequency should be 0.8 every generation. So as we look at the next generation of flowers and the next generation of flowers and the next generation of flowers in that population, we should be able to calculate that the big R allele is at 0.8. If it's not, if it starts to change, if maybe in the next generation it's 0.6, 0.5, 0.4, we're seeing a difference in that every generation, then we know that there must be an agent causing some kind of evolution to take place. So then we take the Hardy-Weinberg theorem and the calculations that we're getting ready to go over to try to determine what is the agent that's causing the change. And we'll look at what the possibilities are. We start weeding out different things that might be causing evolution to happen until we find the agent that's causing that change to take place. So if our gene pool is the big R allele is 0.8 and the little r allele is 0.2, and I've showed you how that calculation takes place, then using the rule of multiplication and the law of addition, we can determine some other things about this population. We can say, well, the big R individuals in the population, then each additional generation from there, if this is the frequency of the allele, then 0.8 times 0.8 would give us a frequency of big R individuals in the population of 0.64, or 64% of the individuals should be homozygous dominant. We can do then that law of addition down here for the heterozygotes and say, well, that would be a big R little r plus the possibility of little r big R, which are going to look the same regardless of which parent gave them each trait. That would be 0.8 times 0.2 plus 0.2 times 0.8, and that gives us the frequency here of 0.32, meaning 32% of the individuals we would expect to be heterozygotes. And then finally, we can finish this off by saying the little r's, that would be 0.2 times 0.2, which would be 0.04 or 4%, we would expect to be, in this case, white flowers. Now notice we've got some things that we could go back to what we've talked about in genetics and say, well, these frequencies, if this is the total population, then these frequencies should add up to 1. So big R plus big R plus little r frequencies should equal 1 for the whole population. And when we put these numbers in, we do find that 0.64 plus 0.32 plus 0.04 do equal 1. And that's going to be important later on as we use this equation. So we have then the Hardy-Weinberg equation, and we have to come up with an equation that we can use regardless of the traits that we're talking about or characteristics. Up to this point, we've been talking about that population of big R and little r, big R being red, little r being white. But we have to make that more generic now because we have to be able to use it from one generation to the next. So now we start using a new letter. Little p is going to represent the frequency of the dominant allele in the population. And Q is going to represent the frequency of the recessive allele in the population. P plus Q has to equal 1. So if we know either one of these, we can figure out the other one just by simply subtracting it for 1, because the population has to be equal to 1. The frequency does. So, and this is also assuming that we're only looking at a trait that has two possible alleles. In this case, big R and little r is the one we've been talking about. It changes as it gets more complex. We won't really go into that in this course. So when we're looking then at, this is just looking at these new letters now and figuring them out in a population. So you would say P squared, or P times P, would equal the frequency of the homozygous dominant, which we already looked at that on the previous slide. We could say Q squared, or Q times Q, would be the frequency of the homozygous recessive individuals in the population. And we would be left with then 2PQ. Now the reason why that's 2PQ is because if you've got P times Q plus Q times P, and we know from algebra that it doesn't matter which one of those you multiply first, that's really P times Q plus P times Q. Well, PQ plus PQ is 2PQ. So that's where that comes from, is the frequency of heterozygotes. So when we put that then in our equation, remember it all has to add up to equal 1, we get P squared plus 2PQ plus Q squared equals 1. And you might recognize this equation from algebra. So this is the equation then that we're going to use in the Hardy-Weinberg equilibrium to figure out whether a population is changing or evolving or not. Let's finish this up with a discussion about some Hardy-Weinberg assumptions. There are 5 Hardy-Weinberg assumptions that have to take place if a population is in an equilibrium, meaning there's no change happening in that population. The first assumption is that the population has to be very large, meaning that you have to have a large sample size, and we've talked about that with experiments. That's always important. The second being that there's no migration taking place, meaning there's no organisms moving into the population or moving out of the population. It has to be a population that is staying stable as far as people moving in and out. The third is that there's no net mutations going on. Mutations are a change in DNA that would affect an allele expression. So we say that there's no change in DNA taking place, that random mating is taking place, there's no specific preference of one mate over another, and that there's no natural selection taking place, that there's no one trait that makes you better suited for survival in this population than another. So when you start to really think about these 5 things occurring in nature, we don't really expect a natural population to be in what we call Hardy-Weinberg equilibrium, because for all 5 of these to be met, that's actually pretty tough to do. We know that natural selection takes place on a regular basis, so that one's tough to rule out, although it can be from time to time. It's tough to say that there's random mating going on, because in most populations there's some type of preference for mate. It's tough to say no mutations, because mutations do take place on a regular basis, although the majority have no effect. There are occasionally those that do have either a positive or negative effect. To say that no individuals are leaving the population and no individuals are coming into the population, many times for that to happen, there has to be some kind of barrier set up, a mountain range, an island, something like that to take place for there to be no migration. And then in natural circumstances, to find very large populations sometimes is very hard. Some organisms we just don't have large populations of. So these are hard to meet to say that there is no evolution taking place. If any one of these is not met, then the frequency of the genes in that population is going to be changing over time, which is the exact definition that we get of evolution: change over time in a population.